Well, thanks for stopping by. I'm Dave Schuster. I am the director of R&D, the head of R&D for Quanta Biosciences, and I'm here today. I wanted to get you acquainted with a new qPCR cycler that Quanta Bio is selling called the Q. So our introductory slide is sort of why Q, and I hope by the time I'm done, you'll be thinking, well, why not Q? It's a very little machine, very compact size, but delivers amazing results and phenomenal temperature uniformity. It's a 48-site rotary-based device. And the unique feature of this machine is that it heats by magnetic induction technology. So very much like your magnetic induction cooktop or those heat plates that you can buy, that's how this heats. And then for cooling, there are fan-based systems that then bring the temperature down. It has ultra-fast data acquisition, which delivers unrivaled performance, really. Everything integrates into a system, very small. It connects to the computer by Bluetooth. You can daisy chain 10 of these machines into a single laptop for operation. So let me take you through some of the principles of it. Again, as I mentioned, it heats by magnetic induction technology. So the rotor being used where that contains the samples. Where's the pointer? There we go. As this spins, it goes through this magnetic coil, and that then induces the field, and the rotor gets hot with no loss of energy. So there's no need for a lot of insulation around this. And again, as it then cuts off the magnetic field, the rotor cools based on just fans. So it's very reliable, very robust. There's no temperature drift over time, and it provides, again, very, very high site-to-site uniformity. So unlike a block-based cycler, where you're having to have overshoots and undershoots, there is no edge effect on this machine because everything is continuous and the tubes are contained in this rotary heat block, if you will. Okay. So what I've included on the bottom are actually heat maps that were done on a Peltier-based machine, which is one of the more precise machines designated for like high-resolution melting applications. And by putting in basically a DNA probe in every well and then looking at a very fine level melt curve on those, you actually have a site-to-site thermometer in every reaction, and you can see that you have differences on a 96 well block, typically of around 0.3 degrees, quite a range. You see profound edge effects on these. But on this machine, you don't have that. And then also coupled to this, on a lot of other machines, the other thing that causes errors in qPCR is your synchrony between data acquisition and sample temperature. Because this rotor is spinning quite rapidly, you have 4 detectors that are for 4 different channels that are fixed in the cycler. And so it's the sample moving across the detection field that then allows for the data acquisition. So on all 40 well site, all 48 samples, under a second of full data acquisition. So, and then the channels are set up to handle some of the more popular dyes for probe-based chemistries and enable quadruplexing. The other part that's unique is really the reaction tube that is used for the device. It can be a bit of a mindset change. It was for me initially when I started working with this, but having the tubes arrayed in a typical fashion, and then the software is designed so that you can load these and it'll translate the sample ID to the proper location in the rotor. but it's the tight fit. There's oil over this, so when you add the sample and there's a cap that goes on and then you spin it, the sample becomes overlaid with oil, very much like the old days, the original days of PCR, but it provides for much tighter, there's no evaporation, and much tighter sample volume uniformity from sample to sample. And so if you add all these components together, really what enables an effective qPCR machine is maintaining synchronicity between sample temperature and your calculated block temperature. With standard block-based machines, you have this asynchronous nature because, again, the block is having to overshoot and undershoot in order to ramp the cycle, the temperature on the sample up and down. You have perfect mating of this on the Q. So one application that you can look to if you're looking to, that demands very high precision of temperature control in PCR is high resolution melting. And so Quanta has a product for high resolution melt called the AccuMelt HRM Supermix. This is a model system composed of 4 separate oligonucleotides with a single base variant in the middle. So we can look at basically all 4 homozygotes allele independently or mix these in different ratios. So if we're looking at the T allele, you can see we end up with a melting temperature of 83.61. This is the A allele. So basically, if we were looking to do typing of an AT class 4 SNP, the temperature control is precise on this machine that you could actually do this just by looking at a melting curve without any of the fancy data normalization required for HRM analysis on typical block-based cyclers. And a similar situation looking at a class 3 GC SNP. Now, if you do get the HRM module, there are other classic normalization routines that are included for this, for normalized melt curve or difference analysis, and you can see the 4 alleles readily pop out. Now, how does this temperature uniformity translate into your more routine use of qPCR for relative gene expression analysis? And in this case, this is just an example of a 2-step RT-qPCR. cDNA synthesis was generated with our QScript cDNA synthesis kit. And then we looked at a dilution of that and amplified product with CyberGreen Fast Mix. So using pretty much just the standard cycling conditions that we recommend for the product. Now this is a compilation of data from 6 different runs. You can take data and you can merge them into a single file, and a total of 21 different replicates, and you can see the very tight precision around that CT call. So that tight precision of data enables much higher accuracy and fine level discrimination when you start to look at 2-fold differences or even down to 1.2-fold differences in input quantity of your template. And it can sustain this again, not just within one run, but you can bring import data from multiple runs and you will see the same kind of tight clustering of data and performance from this machine. Just looking at then, we wanted to know how fast could we push this. Typical cycling conditions for CyberGreen Fast Mix are they're reasonably quick using a 15-second annealing. This is a 3-step protocol, so it comes in about 48 minutes for a 40-cycle run. This is just a dilution series of a linearized plasmid DNA. I believe this was for ACT-B. Some of this data you probably can't see, but the slope for the standard cycling is like minus 3.503. If we shorten the dwell times and augment some of the temperatures here, we can do a 2-step cycling in roughly 30 minutes, and our efficiency is like minus 3.554. So very, very comparable performance whether you're running in a very fast mode or in a standard mode. This just, again, kind of extends it to 2 other assays. For GAP-DH, we look to see how fast we could push it, and we could get GAP to go down in a 20 microliter reaction volume to about a 28-minute runtime. If we further reduce the sample volume so that you have more rapid sample equilibration for BCL2 was an assay that we could push down to about 21 minutes, again for 40 cycles, and this includes the dissociation or melt curve at the end of the qPCR. And you can see how tightly clustered all these replicate reactions are. So as a more general guideline though, if we looked and we surveyed a broader range of genes of interest of varying GC content and length, in a 20 microliter reaction, I think you could safely get by using 5-second denaturation and 10-second annealing extension for a 37-minute runtime. Going to lower reaction volumes, of course, it then allows you to use more sites, so you can daisy chain more of these machines together to collect more relevant data points for you. But you can see we can get down to close to a half hour. This is an example of the multiplex capabilities in a one-step RT qPCR. So this is just a co-dilution of human universal reference RNA with then amplifying with 4 different Pacman-based assays with FAM, CalFluor-Orange 560, CalFluor-Red 610, and the Quasar 670. And you see very nice clustering, tight data across the board, good dynamic range, and bringing in a multiplex one-step run in under 40 minutes. Again, another application just using an off-the-shelf assay. This is just a typical Pacman endogenous control, available from Applied Bio for RPLPO. And you can see that if we use the standard cycle times, with taking about an hour for a run to complete, we can cut that almost in half with absolutely no loss of data integrity or precision in the assay. So with that sort of precision and temperature control, we were kind of positing internally, well, what applications may really benefit from this? And one that jumped out would be using the cycler for NGS library quantification, particularly in, I would say, low to medium throughput labs. I mean, if you're in a lab where you're running 400 libraries and constructing 400 libraries a day, I mean, it would be feasible for this, but you would need, I think, automation and higher capabilities. But this run-to-run uniformity and temperature control, I think, has significant advantages, particularly for this application. A qPCR is generally regarded still as the gold standard. There are a lot of people just getting by using qubit or total DNA quantification. But depending on the integrity and quality of your library or the input starting amount, many people still find a need to use qPCR and to specifically amplify from the P5, P7 sequences in the adapters used for Illumina library workflows. So while it is a gold standard, there are some, I would say, challenges to using the technique for library quantification. Your library length, you've gone from very, very short amplicons, typically used for qPCR experiments, now to libraries with average length of about 450 base pairs. You're needing to accommodate varying GC content. Typically, on a plate-based instrument, you need to include all your standard DNAs on that plate, so everything is using absolute quantification within that run. Your library itself, you need to often make have multiple dilutions of that, and there are inherent errors associated with that dilution process, such that your sample falls within the linear dynamic range of your standard curve if you're going to use the technique and achieve accurate quantification. So with the Q, we think we can simplify library quantification, mostly by preserving this run-to-run uniformity. So we're able to shorten run times. Doing so, we can reduce operating costs. And by preserving run-to-run uniformity and using a single standard curve across multiple runs for different libraries, it opens up greater real estate for for your library samples and reduce operating costs. The broad dynamic range of the instrument kind of reduces some of the the need for multiple dilutions. And again, by setting up one standard curve and then applying samples to that, because we don't have inter-run variability, we can shorten the workflow. So the current kit that we have on the market for NGS library quantification is called the Perfecta NGS Quantification Kit. This is targeted for Illumina chemistries. Again, contains Perfecta Cybergreen Supermix. On block-based cyclers, we found this particular master mix to be a little more forgiving, enable longer amplification. It was more forgiving. The kit also contains stabilized pre-diluted standards. We've left these sit on the bench for as long as 6 months, and we see no degradation in CT calls for these DNAs. And then we also include the same stabilization buffer that we use for the standards to dilute your library in. Now the downside is, in order to accommodate libraries of varying length and varying GC content, we opted for a very conservative, rather slow protocol. So it's about an 85-minute runtime from start to finish to then get your answer. But this shows the high reproducibility of these standards on the queue. These are 4 independent runs with 3 technical replicates for each standard DNA concentration that are then compiled into a single project on the Q qPCR software. And you can see there's basically no drift between them. Now this may be a little hard, and this is a lot of data to dig through, but what we included in this run too was a sample library. Now the library is created with our Spark NGS library workflow. It consists of a mixture of 3 bacterial genomes from Francisella, Escherichia coli, and Bordetella. So we're spanning a GC content range of 8% all the way to 90%. And after the enzymatic fragmentation, it has about an average fragment length of 450 base pairs. So we use this material, we have this highly standardized, we know the concentration's been verified by a number of independent methods. And so we wanted to see how this sample would then be quantified both within each run and by applying the standards from separate runs. And you can see that you have roughly the identical quantification call regardless of which set of standards you're using from which run to make a call on this particular library. So basically, DNA standards, I think the data demonstrates that standards are not needed in every run. Again, that frees up more space for more library samples and then can lower operating costs for you. Now it's still taking time to do this. This is still using the standard cycling conditions that we recommended, but under those cycling conditions, you can see that we can readily resolve between 1.5 and even get down to 1.2-fold differences in input quantity. So we can achieve very accurate results. And again, this is using that more challenging library model system. So we then wanted to see how fast with the machine capable of pushing the technology. And the best we could do with the Cybergreen Supermix was to cut this from about 85 minutes down to 50 minutes, which is still a significant savings. You're saving almost a half hour, and with no loss of data integrity or quality by doing so. And lastly, we went back and wanted to revisit Cybergreen Supermix. It was just a bit on a whim. When we initially developed the kit, and again on these block-based cyclers, we found that the fast mix, for whatever reason, delivered very poor results, even with standard cycling conditions on block-based machines. But Peter Bartholomew took this on, and this is a log-fold dilution of that library, model library, and comparing results between the classic standard cycling conditions with Cybergreen Supermix and highly optimized rapid conditions with Cybergreen Fast Mix. You can see both give very broad dynamic range, have roughly equal efficiencies, and so we are actively looking at developing now a new kit specifically for the Q with a highly streamlined workflow that will simplify your operations and make library quantification easier and hopefully faster. And CyberGreen FastMix is really just our first look at this, kind of demonstrating proof of principle with the capabilities within the team, within the R&D for protein engineering. We may be looking at faster polymerases and even be able to reduce this and to improve the quality of this even further. So this just sort of shows you the time savings that can be achieved, either using the typical library quantification cycling protocol, optimized protocols with the existing kit, and then down to cutting it down to about a half hour. So we're going from an hour and a half to about a half hour with CyberGreen FastMix. So hopefully I've convinced you somewhat you could give the kind of the pun is intended, give the Q a spin. I think you'll be very satisfied with the results. It's a machine that once you start using it and you get familiar with it, you like it more and more every time you use it. And I'm telling you that as someone that's actually used it personally. Collectively, when you look at the components of the system, the magnetic induction technology, the high temperature uniformity, the rapid optical data acquisition, and superior data analysis algorithms, I mean, most of what works best on here is utilizing the LinReg program for CQ calls. Altogether, you get superior data analysis. You have, combined with the tubes, these work as a system, and then they're reagent agnostic, but they're compatible with a wide range of reagents to deliver, I think, just the best quality qPCR performance with a wide range of applications. Now, specifically with regard to NGS library quantification, we've demonstrated that we can obtain highly precise measurements over multiple trials. So we can streamline some of that workflow by using single set of standards over multiple runs. We can achieve high amplification efficiencies with a wide variety of cycling conditions. And we're looking to push this even further, into saving even more time with new optimized kits to basically make your life easier, allow you to get reduced time to result, and to get your samples on the machine, on the sequencer, faster and obtain more reliable data for your studies. So with that, I'd like to acknowledge Peter Bartholomew. He has a poster over here on the NGS library quantification. I encourage you to go visit that and talk with Peter. He has done all these applications work on the team. My other R&D team is divided into two groups. Reagents R&D are working on workflows for both qPCR, PCR, and NGS workflows. And both Hanbo Liu and Yijin have workflows, posters on different NGS workflows here. On the protein engineering side, Tom Schoenfeld leads that group, and they're presenting a new enzyme, which is a thermostable reverse transcriptase, which again is something that we hope to deploy for very robust, reliable one-step RT-qPCR applications on the Q. As I said, it's a very small, it's portable. We've had, with the robust nature of like qPCR tough mix, we've had researchers from CDC take this into the Rift Valley and get exceptional performance in the field with this little box. We're hoping to do the same with one-step, solutions for these challenging applications. And then we never would have got this on board without our marketing team led by Heather Meehan, Brian Komros, and Eleanor Postolowski. Well, thank you for your time and attention. If you want to take a closer look at it, please stop by the booth and also check out the posters from the other members of my team. Thank you.